Good morning, dear students. We are back with the second year microbiology part of immunology, and today we are going to consider a part called as antibody structure and their different functions. So, what are the different functions of antibody? Last lecture was with antigen. Now this lecture will be of antibodies. So let us understand. Try to go along with this particular part. Now immunoglobulins, so antibodies are also called as immunoglobulins. Okay. So why they are called as immunoglobulins? When structurally we are going to see this part, at that time we will come to know that why they are called as immunoglobulins. So these are glycoproteins which serum portion of the blood, present as glycoproteins inside the blood serum. Okay. It's composed of 82 to 96% polypeptides and 2% to 14% of carbohydrates. Humoral branch of immunological response. See, there are two basic branches of immunological response. One is called as the cell-mediated immune response, other is called as humoral mediated. Whenever you will heard a word called as humoral immune response, at that time it will be associated with immunoglobulins or antibodies. Very good. So, play role in antigen recognitions and biological activities related to immune response and complement activations has 5 major classes. So, it has 5 major classes. Whenever there is an infection inside the human body and whenever there is a counteraction of the antigen and antibody, then and only then a positive immunological response can be mounted. So 5 different types of immunoglobulins are there: IgG, IgM, IgA, IgD and IgE. So what are their functions and all? We are also going to deal the same part. Next. So immunoglobulin structures are tetrapeptide structures. Interchange disulfide bonds create flooded regions or domains. The amino terminal lengths of each chain is a variable region, while the carboxyl terminal length is one of the more constant. Peptide digestion yields FAB fragments, while all antibody activities are on FC fragments. Peptide digestion yields 2 FAB fragments and on FC portions. So antibodies are what? These are interlinked with tetrapeptide structures. So structure, if we see this. So this is structure of the basic antibodies. The antibodies may have heavy chains. These are called as heavy chains, and these are light chains. Okay. So when peptide digestion will take place, it will act on this hinge region. Either it will be cutting the antibody into 2 different parts like this, or it will be cutting the antibody into FAB and FC portion. Okay. So this will be FC portion. This will be FAB portion. 5 different structures of antibodies you are going to see over here. Of course, in the next lecture we are going to see what are the different structures of antibodies. For the time being, these are 5 different antibodies, that is IgA, IgM, IgG, IgE and IgD. Okay. This is peptide digestion. Peptide digestion, it will digest or it will cut this particular part into 2 parts. So that will be helpful for us for disintegration of FAB and FC portions. Now, nature of the light chains. The light chains have kappa and lambda. Constant regions, C-terminal region ends and similar amino acid classes of antibodies. Variable region includes 110 to 130 amino acids of the light chain and the heavy chain responsible for binding of antigens. So this is one of the most important part of variable region, because there are number of antigens which are present. In order to recognize them, there is a sequence of amino acid up to 110 to 130 amino acids are present over there, and then it will be working it out. Heavy chains will be gamma, mu, alpha, delta and epsilon. So based on the heavy chain, classification of immunoglobulins has been done. We can easily see that how IgG, IgM, IgA, IgD and IgE have been easily been classified. Isotypes, unique amino acid sequence that is common to all immunoglobulin molecules of a given class in a species. Allotops found in the constant region are inherited in the simple Mendelian fashion. Occur 4 IgG classes in or 1 IgA subclass in the kappa region. Idiotype found in the amino acid terminal of both L and H chain, essential for formation of antigen binding site. Together they survive antigen recognition unit. It means that there are certain regions inside the antibodies also which are said to be Either it may be isotype, allotype or idiotypic type of antibodies. It means that some of the antibodies may be inherited by our mother, or from our mother we are getting those particular antibody. So the amino acid sequence will be very much similar what our hereditary characters would represent. Isotype, allotype, hinge region. The flexible amino acids stretching in the central part of the heavy chains of IgG and IgA immunoglobulins classes, which links these two chains by disulfide bridges. See this. This is the hinge region. Okay, so this is the hinge region, what we are talking about. Rich in cysteine and proline amino acid, extremely variable in the amino acid sequence, no resemblance to any other immunoglobulin region. Gamma, delta and alpha chains have a hinge regions, mu and epsilon doesn't have any sort of hinge regions. Now, this is the 3D structure of antibody. You can see these are the disulfide bridges which are attached over it. Fructose moiety which is present over here. N-acetyl D-glucosamine, the sugar. Alpha D mannose here, and D-galactose, again a sugar which is present over here. Okay, so this is nothing but glycoprotein molecule. Why glycoprotein? Because it contains glucose molecules or sugar moieties as well as proteins. Okay, so what we have learned in this particular part today, that there are 5 different types of antibodies, and these antibodies have different functional role to play. In our next lecture, we are going to concentrate on what are the different types of antibodies and what are their functional role. Correct? This was the very basic lecture which was associated with what we can say with the structure, basic structure of an antibody. There will be variation in the structure of the antibodies when it will be covering what we can say the IgA, IgG, IgD, IgE and IgM, different types of antibodies we are going to learn. At that time we will come to know that what are the different structures and functions of this particular antibodies. Correct? So that's all for today. Thank you.